My name is S. Ashok Kumar. Welcome to my channel. In the last part, I explained about the metabolic pathway of Krebs cycle and its importance. Now, in this part, I am going to explain bioenergetics of aerobic cellular respiration per glucose, or you can call it as exo sugar. Here, I am going to tell about in which reaction mediated by the corresponding enzyme, energy, whether it is released or consumed, now I am going to tell. As overall, we can calculate how many number of ATPs produced. Okay, na? The first reaction itself, that reaction is mediating by enzyme hexokinase. In this reaction, hexokinase reaction, there is investment of 1 ATP. That is why I am calling as a minus 1 ATP. Minus means what? Utilization, not release. Utilized. The next reaction mediating, that is a phosphofructokinase. In this reaction also there is investment of 1 ATP. If you want to understand this bioenergetic, you should read glycolysis and Krebs cycle. Okay, na? The next reaction mediating by the enzyme, that is a glyceraldehyde 3-phosphate dehydrogenase. The reaction mediating by this enzyme, that is the first oxidation step. There is a release of 2-NADH2. So when these 2-NADH2 involved in the electron transport chain, how many number of ATPs produced? That is a 1-NADH2 gives the 3 ATPs. Okay, 2-NADH2 gives the 6 ATPs. So plus 6 ATP. What is the plus indicate? That is releases. Minus means utilized. The next reaction, that is mediating by the enzyme bisphosphoglycerate. Bisphosphoglycerate kinase. This is the first SLP. Here there is a release of plus 2 ATP. Okay, na? So here I am writing this is the first oxidation. In this way, so you can remember. This is the first SLP, substrate level phosphorylation. Next reaction mediating the enzyme pyruvate kinase. This pyruvate kinase, in this reaction there is a release of 2 ATP. This is a second substrate level phosphorylation. Okay, up to here glycolysis. Next one is what? Pyruvate enter into the mitochondrial matrix where it converts into acetyl-CoA. So that is a link reaction mediating by the PDH complex, pyruvate dehydrogenase enzyme. Here there is a release of 2-NADH2. When they involve in the electron transport chain, cell will get the 6 ATP. This is an example of first oxidative decarboxylation. Already I mentioned. Next reaction, then acetyl-CoA formed, then enter into the Krebs cycle. Next reaction, that is a isocitrate dehydrogenase enzyme. Here also there is a release of 2-NADH2 as we know. It gives us 6 ATP. This is a second oxidative decarboxylation. The next 8th reaction mediating by enzyme alpha ketoglutarate dehydrogenase. Exactly like there is a release of 2-NADH2 that produce 6 ATP during the electron transport chain. This is an example of third oxidative decarboxylation. Okay. Next reaction, that is a mediating enzyme succinyl thiokinase. Here in this reaction there is a formation of 2 GTP. 2 GTP is equals to 2 ATP because GTP and ATP are equivalents. Here this is an example of third substrate level phosphorylation. Okay, na? Next reaction, that is a succinate dehydrogenase. Here there is a release of 2-FADH2. Okay, na? Following. When one FADH2 involved in the electron transport chain releases the 2 ATP. So 2 FADH2 gives the 4 ATP. 4 ATP. This is an example of second pure oxidation. No decarboxylation, only oxidation. This is the first oxidation. The next one is mediating by enzyme, that is malate dehydrogenase. Here this malate dehydrogenase enzyme produces the 2-NADH2. 2-NADH2 gives the plus 6 ATP. This is a third oxidation. Now this you can go for the net. Here see this one first. How many are produced? First you count. This is a 6 ATP. 6. Then 2. 8. Then 10. Okay na? 10. Next 16. 16 plus 6 how much? 24. Yes or no? 24 6 how many? Sorry, we will count one more time. This is the 6. Then 8. This is the 10. Okay na? 10 plus 6 how many? 16. 16 plus 6 how many? 22. 22 plus 2? 28 na? 28 30. 30 plus 4 34. Then 40. That means the total number of ATPs produced per glucose 40 number. But in the initial step of the glycolysis, 2 ATPs are utilized in. So we have to remove the 2 ATPs from the total. That is why we will get 38 number of ATPs per glucose or per hexose sugar. This is so the number of ATPs produced during the aerobic cellular respiration per glucose or per hexose sugar is equals to 38 number of ATPs. This is about the bioenergetics of aerobic cellular respiration of glucose or hexose sugar. Explaining the bioenergetics of the Krebs cycle, that is the aerobic cellular respiration that include both glycolysis and Krebs cycle. Now I am going to explain about the anabolic nature of the Krebs cycle. This is roughly I showed some of the intermediates of the Krebs cycle that as we know. First you take the acetyl-CoA. This acetyl-CoA involved in the synthesis of carotenoids and gibberellins. Gibberellins means carotenoids and gibberellins are synthesizing from the ester with involvement of the acetyl-CoA. Next you take the oxaloacetic acid. Simply I showed the OAA. Oxaloacetic acid by undergo amino transfer, that is a transfer of amino group that involve in the synthesis of aspartate. What is aspartate? That is one of the non-essential amino acid. You remember oxaloacetic acid involved in the synthesis of aspartate. This aspartate involved in the synthesis of other non-essential amino acids. Non-essential amino acid. As we know, this aspartate is a standard amino acid. But out of how many types of standard amino acid? 20 types of standard amino acid. Out of 20, 10 R belongs to essential, 10 R belongs to non-essential. Then now comes to the citrate. Citrate is very important intermediate that involve in the synthesis of fatty acids and cholesterol. Definitely cholesterol is absolutely required. Next it takes the alpha KGA, the only 5 carbon molecule formed during the Krebs cycle. Like oxaloacetic acid, if you clearly alpha ketoglutaric acid by undergo aminotransferation or amination, that is a transfer of amino group, this alpha KGA is converted into glutamate. What is a glutamate? Glutamate is also one of the standard amino acid This glutamate in turn involves the synthesis of different types of non-essential amino acids. You can understand why they are called as a non-essential, because these amino acids no need to supply through the diet, because those non-essential amino acids can synthesize in our body cells. Next comes to the succinyl-CoA. Succinyl-CoA involved in the synthesis of heme. We heard this a number of times. This heme is a part of hemoglobin, that also part of cytochromes. Cytochromes are very important of the electron transport chain. Now you can understand the anabolic nature of the Krebs cycle. Here what I said, with this one what you need to understand is, some of the intermediates of the Krebs cycle involve in the synthesis of different types of substances that are required by the cell. That is why this Krebs cycle is having the anabolic nature, because some of the intermediates involve in the synthesis of other required substances. This is a one. In the Krebs cycle, already I said, whatever the acetyl-CoA involved in the Krebs cycle release in the form of carbon dioxide and generates the energy. That is a catabolic nature. So that is a catabolic nature. Why Krebs cycle is called as a catabolic nature? Because Krebs cycle is responsible for producing the most of the energy. That is a catabolic nature. Why it is a Krebs cycle is also having the anabolic nature? Some of the intermediates of the Krebs cycle involve in the synthesis of required substances within the cell. That is why, as a conclusion, what you can tell? Krebs cycle is having both catabolic and anabolic nature. That is why Krebs cycle is an amphibolic nature. What is amphibolic nature? It has both catabolic nature by supplying the energy, and also anabolic nature involving the synthesis of different types of required substances. That is why I am concluding the Krebs cycle. Krebs cycle is an amphibolic pathway or an amphibolic nature. So till now, if you understand the concept, you can like and share this video to your friends and subscribe our channel. If you have any doubts about the chapter for your NEET exam, you can comment below. Definitely I will respond to your questions. Thank you.